Is the diarrheal disease cholera and the bacterium that causes it, Vibrio cholerae. And here you see a 19th century representation of King Cholera, the king of diseases, a disease that, not surprisingly, killed a lot of people prior to proper sanitation and toilets and so forth. And so here's the cholera king feeding individuals in this poor neighborhood contaminated water from a well, from this pump that becomes central to a story that of cholera that occurred in central London during the 1800s. So in this particular breakdown, we look at some of the history. There were 7 major pandemics since 1817. The first 6 arose basically in the Indian subcontinent and spread to other continents. Not surprising how that could happen, right? Because India was colonized, for example, by Great Britain, and so there was a lot of exchange of people and cholera between the two places. The first 6 were caused by Vibrio cholerae, serotype 01, biotype classical, and I'll explain that in a minute. And the 7th, there was a shift in the biotype from classical to Altor. So there is a neat little ecological succession happening where different strains are kind of competing for dominance in the world stage. The 7th pandemic was caused in Sulawesi, Indonesia, and then spread to the Asian mainland, and then from the Asian mainland it spread to Africa, and then ultimately to South America, and caused a lot of death and destruction. And then in the 90s there was a huge outbreak of cholera in Zaire, which is now the DRC, or Democratic Republic of the Congo, and there were 12,000 Rwandan refugees that were killed. And this is primarily because of a cultural clash between groups Hutu and Tutsi. We don't need to get into the details, but the point is that because of this sort of ethnic clash, these individuals, the warring that was going on caused a lot of people to flee Zaire into neighboring Rwanda by the tens of thousands, and they set up these massive camps, and obviously cholera, which is a disease of misery, just ripped right through them. If you ever get a chance to watch the movie Hotel Rwanda, there's a bit of the backstory and basically explains a bit of what happened during that time frame. I don't think they specifically get into cholera, but they do get into the brutality that led to the Rwandan refugee crisis. And so, a nano 1 strain of cholera, so a different serotype 0139, became prevalent around the world and possibly is causing an 8th pandemic. And we see cholera outbreaks, for example, happening right now in places like Yemen. And again, it's a disease of misery, so it's following the civil wars and so forth that are happening in other countries, particularly poor countries. This slide is just a representation of the different serotypes of cholera. And so we start with Vibrio cholerae, and then the 01 serotype, and then the 2 biotypes, classical and Altor. Don't worry about these guys in the middle. They're typically we just lump them together in this nano 139 group. And then the other pandemic strain of cholera is 0139 Bengal. So 2 serotypes, 01 and 0139, are causative agents of cholera outbreaks. And so the serotyping is done by looking at the so-called O antigen on the surface of the bacteria, and that's these are gram negatives, and so only gram negative bacteria will have this O antigen, which is basically just a lipopolysaccharide with a sugar side chain. So that's this material right here. So the lipopolysaccharide, or LPS, portion of the outer membrane, which is also referred to as endotoxin. But the different sugars that form these little branches can be used to serotype the microbes and lump them into different groups, O1, O2, O3, etc. Another way we serotype microbes is through their K antigen or capsular antigen, so different sugars that make up capsules can be used to serotype. Another one is H, so the flagella antigens that are on different microbes. And then F can also be used, an F antigen for Fimbria. But in this particular case, the O1 refers to the one type of lipopolysaccharide side chain. O139 refers to the 139th version of it. So just getting back to that historical thing that I'd mentioned, Jon Snow, classic epidemiological study on cholera. So this is 1854 in London. And so Jon Snow, who was a physician, was tasked with trying to figure out the cause of the cholera outbreak that was happening at the time. And Snow hypothesized an association of cholera with drinking water in this particular epidemic, which ultimately encouraged what everybody hailed as a major event, was the removal of the pump handle. And so how did he do that? Well, one of the great things about this story is it represents the first time where science and sort of this new field of epidemiology was applied to understanding outbreak dynamics. And so everybody these days with COVID-19 is becoming an epidemiologist, with all the terms floating around, R naught and CFR and IFR and all these things. So it's interesting to see how far we've come. But this, again, was the first time that individuals had really applied a sort of a science-based approach to understanding where the outbreaks are coming from, because a lot of this outbreak studies and things were based on sort of mysticism and old beliefs. And so this particular outbreak, for example, was believed to be caused by the bad smells coming out of the river. And associating smells and toxic fumes with disease is not outlandish, right? Because even Jon Snow was an anesthesiologist and understood that you could knock people out with chloroform and stuff. So it wasn't that hard to imagine that some foul stench coming from the Thames River could be causing cholera. But Snow didn't like that idea, because how could the cholera be so focused in certain areas and not others if everybody's blanketed in this foul smell? I think you got to remember that London was a very filthy place back then, and a lot of our books and stories always imagined people in top coats and everybody in fancy clothes, but the reality is London was largely just poor people, and with a very, very narrow range of extremely wealthy people. So wealth distribution wasn't great. There was horse manure in the streets and all that. So anyway, so what Snow did essentially is create a map of the regions of London, and basically looked at deaths essentially per household. And so each of these little black bars represents a death in a particular household. So here's 3 different houses, and then the pile of deaths in that particular household. And you can see some of the houses were really affected, but obviously these are in many cases apartments, where hundreds of people lived. But by doing this approach, was able to come up with a sort of a map, a death map, a ghost map. There's actually a book called The Ghost Map, which is really recommended reading. But anyway, the idea is, looking at where the cases sort of, where the deaths sort of are most dense. And he found in this particular area, there was a lot of deaths surrounding in this particular region of town on Broad Street. And right in the middle here is that pump, the Broad Street pump, which leads to a well below ground that individuals drew their water from. And that led Jon Snow to the conclusion that the pump is probably distributing contaminated water. And so you have to remember that this is the 1850s, and Robert Koch and Pasteur had not yet elaborated the germ theory of disease. So again, there was that association between microbes and disease hadn't really been made yet. So that's why Jon Snow said, let's knock the pump handle off, prevent people from getting water from this particular location, and force them to go somewhere else to get their water to the other pump. And as the story goes, the deaths and cases declined rapidly thereafter. But the reality is the cases were already on their way down, and deaths were already on their way down. So the infection had already kind of pushed through the population and was on its way down. But that doesn't mean that Snow wasn't correct in his hypothesis that the pump was the problem. And so later on, Robert Koch got involved, and so he isolated the Vibrio cholerae, as he called it, the Kama bacillus, from patients in a cholera outbreak in Egypt, and then used his postulates to attempt to relate the presence of this Kama bacillus to the watery diarrhea that's typical of the disease. So some of the characteristics of cholera, it's a gram-negative bacterium, as I already alluded to, because it has that O antigen side chain from LPS. It's sort of comma-shaped, as you can kind of see here in this electron micrograph. It's a little bent rod. And it's a facultative anaerobe. So again, the microbe can grow with or without oxygen, but prefers to be in the presence of oxygen. So it's metabolically versatile. It's motile by a single polar flagellum, as you can see here, which is very powerful and gives this guy the ability to swim quite fast and get through mucus, for example, in the small intestinal lining, so that it can set up shop on the epithelial cell surface. The microbe grows in salt and fresh water, which is unusual for Vibrios. It doesn't have a real sodium requirement, so this gives it the ability to, contaminate a river, contaminate an estuary, and not quite an ocean. It doesn't prefer that, but it can kind of get into and grow in brackish water. It has poor viability at low pH, which is kind of paradoxical because this is a microbe that has to get through your low pH stomach, and it's much more tolerant of higher pH, and so that's interesting. But obviously, if the microbe is eaten in food or something that protects it, or if it just comes in such huge numbers that even if a bunch of the cells die, all you need are a few to get through to the duodenum and ultimately the small intestines. So there's about 150 serotypes. I've mentioned that O1 and O139 are the bad guys. And by that I mean those are the toxigenic strains. And I've already mentioned that there's two biotypes of O1, L-Tor and classical. The fundamental difference between them is that the L-Tor strain produces a hemolysin, but that's a little murky. I mean, the microbes are constantly sharing DNA and all that, and so it's probably not that clean anymore. That it's biotype L-Tor that just produces hemolysin. I wouldn't be surprised if somebody isolates classical strains that also produce it. So the bit of characteristics of Vibrio cholerae. The organism has two chromosomes, which is unusual. A lot of bacteria have just one, a single, circular DNA, double-stranded DNA chromosome. But Vibrio bacteria have two chromosomes. The major virulence factor is cholera toxin. So if you take away the cholera toxin genes in Vibrio cholerae, their virulence goes way down. The cholera toxin gene, or CTX, expression requires TOX-T and TOX-R, so two transcription factors, and I'll show you that. And the cholera toxin genes are located, there's two of them actually, the CTXA and CTXB, and they work together, and I'll explain that soon, are located on the genome in an integrated bacterial virus, which is really cool. So the bacteria only become toxigenic after being superinfected with a virus. And that's a common phenomenon in microbiology, the fact that viruses can bring toxin genes to microbes to make them deadlier. The virus that infects cholera the cholera bacteria is called CTX-Phi, or this is the Greek letter phi, or phi as the fraternities like to call it. But CTX-Phi is the bacterial virus, and it requires this particular receptor, the toxin co-regulated pilus, in order to infect the cholera bacteria. The other point to make here is that the toxin co-regulated pilus is encoded by a VPI, or Vibrio pathogenicity island. So this is just a hunk of genes that the bacteria acquire through horizontal gene transfer. And the genes on that pathogenicity island can impart all kinds of virulence factors. So this is just looking at the process of transduction. So transduction is the ability of viruses to transfer DNA between hosts. And so this is what gives rise to the toxigenic cholera strain. So here's a bacterium with its prominent toxin co-regulated pilus. And again, the bacteria normally would use this TCP to bind to intestinal epithelial cells so that they won't get washed away when you have bowel movements and stuff. They can actually attach and stay fixed. But the virus can use it as a homing beacon to find the bacteria. And then so when it infects, here's our virus right here. When the virus infects, it injects a single-stranded positive single-stranded DNA into the cell, and then that cell or that single strand of DNA is, as you see here, is circular and can then be made into a double strand. So it behaves kind of like a small extra chromosomal piece of DNA, like a plasmid, for example. The plasmid then can be used to make more genomes, which can then be packaged in the budding virus. The plasmid can also be transcribed into messenger RNA and translated into phage proteins, which again are needed to make the virus structures. And the other thing that happens is the phage DNA integrates into the genome of the bacterium. And that's what's happening here. You see this little crossing over event where this virus DNA enters the bacterial chromosome and then becomes stably incorporated, as you see here. So the bacterium is not killed by this process at all. It actually, accepts the viral DNA and then replicates it along with its own chromosome. And periodically will start to express that material so that over time viruses can be extruded from the bacterial membrane. And so by that process, toxigenic Vibrio cholerae strains can contaminate contaminate the environment with more of these viruses, which can then spread the toxin gene to more bacteria. And this is just showing you Vibrio cholerae genome organization. So here are the two chromosomes that I talked about. And the main things to look at are these over here, these two here. Don't worry about this, this integron thing. Don't worry about it. As cool as it is, I'm not going to hold you to knowing about it. But these two I want you to know about. So VPI is the Vibrio pathogenicity island, and it exists here in chromosome 1. And VPI actually encodes TCP, the toxin co-regulated pilus genes, and TOX-T, which is a transcription factor for cholera toxin expression. Here's where CTXV DNA integrates into the chromosome, somewhere over here. And in this particular case, the genes on VPI, such as TOX-T, can come over here and interact with the cholera toxin genes here, and then give this guy the ability to not only pump out viruses, but also pump out toxin when the bacterium needs to. So the bacterium isn't going to do that when it's floating around in an estuary, but it will do that when it recognizes that it's in a host, so when it's in a small intestinal tract.